Looking at this animated diagram of a chromosome, we can see how the cell organizes its genetic library. The chromosome itself is the storage unit. Inside, we find DNA, the master instruction manual for the cell. To carry those instructions out, the cell prints temporary working copies known as RNA. Notice how this structure locks together into a single unit. Both DNA and RNA are nucleic acids, meaning they are built entirely by stacking millions of these repeating three-part puzzle pieces called nucleotides. Every single nucleotide breaks down into three precise chemical components. We have a phosphate group on the left, a pentose sugar in the center, and a nitrogenous base on the right. Memorize these three parts. This specific breakdown is a high-yield concept for your WBCHSE exam. While these three components are the same across all life, the specific order and chemical combination of these pieces allow cells to store an immense amount of complex biological data. Here we see two distinct pentose sugar molecules. RNA uses normal ribose, but DNA uses a modified version. Look at what happens when we remove one single oxygen atom. The prefix deoxy literally means missing oxygen. That single missing atom gives deoxyribonucleic acid, DNA, its name. Now let's look at the third piece of the nucleotide, the nitrogenous bases. These bases sort naturally into two structural families. We have the purines on the left and the pyrimidines on the right. To lock the purines into your memory, use a simple trick. Think of the phrase pure as gold. Pure stands for purines, the A is for adenine, and the G is for guanine. That leaves the pyrimidine bucket for the remaining bases, cytosine, thymine, and a special base called uracil, which is exclusive to RNA. Whether a strand of genetic material becomes DNA or RNA depends entirely on these specific chemical selections, whether the cell uses ribose or deoxyribose, and exactly which bases are pulled from those two buckets. Let's watch how these nucleotides actually assemble. The alternating sugars and phosphates lock tight to form the rigid outer rails, or the backbone. Meanwhile, the nitrogenous bases point inward, meeting in the middle to create what looks exactly like the rungs of a ladder. The biology of these rungs follows strict rules. Adenine must always pair with thymine, and cytosine must always pair with guanine. Examiners love to test your knowledge on the exact hydrogen bond counts holding these pairs together. Adenine and thymine connect with exactly 2 hydrogen bonds. Cytosine and guanine require 3. Because of the chemical tension created by these specific bonds, the flat ladder cannot stay straight. It naturally coils into the 3D double helix shape we instantly recognize as DNA. Let's test this rule. You are looking at a single strand of DNA reading A, C, T, G. Pause the video right now. Predict the letters of the opposite strand and write down the exact number of hydrogen bonds between each pair. A pairs with T using 2 bonds. C pairs with G using 3. T pairs back with A using 2. And G pairs with C using 3. This mathematical predictability ensures that when a cell divides, it can copy its entire DNA library without scrambling the genetic code. Now look at what happens when we compare this to RNA. RNA departs from the strict double helix shape. Structurally, it operates almost exclusively as a single exposed strand. It also breaks the chemical rules. RNA has 0 thymine. Whenever you would expect a T, RNA pulls from the pyrimidine bucket and swaps in uracil instead. Because RNA lacks a second strand and the rigid hydrogen bonding of a double helix, It is a more flexible molecule that can easily travel to the cytoplasm to build proteins. We are now going to build the exact 4-point comparison table you need to secure full marks on your WBCHSE exam. Point 1 is the chemical difference in the sugar. DNA uses deoxyribose, missing one oxygen atom, while RNA uses standard ribose. Point 2 covers the nitrogenous bases. DNA pairs adenine, thymine, cytosine, and guanine. RNA swaps out the thymine for uracil. Point 3 outlines the morphological structure. DNA forms a double-stranded helix. RNA is single-stranded. Finally, point 4 is their functional role. DNA stays in the nucleus as the master blueprint. RNA travels between the nucleus and cytoplasm to handle protein synthesis. Review this final cheat sheet. Remember, pure as gold to keep your purine straight, and take a screenshot of this frame for your revision notes. Mastering these chemical and structural differences is the first step toward understanding how genetic information is actually stored and translated within the cell.